Good afternoon, all. Thank you for joining us for this free webinar today. My name is Alex Cloherty, and on behalf of Oncode Accelerator, I would like to welcome you to this webinar that we're hosting in collaboration with two expert speakers from Symeres and Leiden University. Before we get into the meat of the webinar, I would like to share a few general housekeeping notes, as well as a very brief introduction to Oncode Accelerator. So first, the housekeeping. This webinar will be recorded and transcribed. So the good news is if you're coming in a little late, you won't have missed anything. You can access the recording on our website afterwards. Throughout the webinar, your microphones will be muted. So we would ask that if you have any questions, please use the Q&A box. You can type your questions at any time, and we'll come back to them at the end of the webinar, during which time is allocated in order to address any questions or remarks that you might have. Now, before I invite our speakers to the stage, a brief introduction to Oncode Accelerator. At Oncode Accelerator, our mission is to collaboratively optimize and accelerate the development of new cancer therapies for the benefit of patients of all ages. And I emphasize the collaborative nature because it's an ambitious mission that no single party can achieve on their own. So this is why Oncode Accelerator brings together over 35 public and private partners from the Netherlands and beyond, including Symeres and Leiden University, who are working together to build and validate a lasting infrastructure to support this mission. Our focus is on specifically innovating and accelerating the preclinical discovery and development of 4 key types of cancer therapies that are represented by our work streams, namely small molecules, biologics, cell and gene therapy, and therapeutic vaccines. And by overlaying our innovation platforms, which are well-defined patient cohorts, organoids, and artificial intelligence, we aim to accomplish this optimization and acceleration of these different types of cancer therapies. To validate this approach, we run demonstrator projects, which are preclinical discovery and development projects that include at least one of the work streams and one of the platforms. Anybody with a validated hit or target can apply, and this way they gain access to our infrastructure, to our innovations, our know-how, as well as co-funding. Now, to make sure that the results of demonstrator projects actually have the best chance possible of seeing the market, we also integrate our regulatory innovation workstream, and this helps ensure that late-stage development criteria are considered early on during R&D. And we envision that the results of these demonstrator projects will prove that it is really possible to accelerate and innovate and de-risk the preclinical R&D of oncology therapies so that we can deliver ultimately effective and safe novel therapies for the benefit of different patient groups. Now, again, the call is open for you to apply. So anyone with a validated hit or target can join us by applying for a demonstrator project, through which you can gain access not only to up to 1.5 million in co-funding, but also project management support, access to our unique research infrastructure, as well as access to the network of world-class experts that are represented across our 35+ public and private partners. To learn more, I invite you to reach out to our business development team at demonstrators@oncodeaccelerator.nl, and we'll pop that in the Q&A as well as it's on our website if you would like to find out more. So now, without further ado, I'm very happy to introduce our speakers and the topic of today's webinar, the future of small molecule-based cancer therapeutics. We'll be led through the core of the webinar by 2 experts from, again, Symeres. Dr. Luc van Hijfte will be bringing the first presentation today, and thereafter, Anthe Janssen of Leiden University will guide us through. Luc, would you like to start to pull up your slides now? And as he's doing that, I would like to give a brief introduction. So Dr. Luc van Hijfte is Senior Vice President at Symeres, as well as sitting President of the European Federation of Medicinal Chemistry. He has extensive experience in pharma and biotech, both at established giants like Janssen and Sanofi, as well as startups like Phytodia. And he's contributed to a wide range of successful projects, including several entering into clinical development. Luc, if you minimize the Teams screen, then we should see your presentation. Little technical. Yeah, that one.
Yeah, it doesn't move.
Oh, how about if you navigate to, yeah, there we go.
I'll get it out of the way like this.
All right. And if it looks to you like you can only see the screen of your PowerPoint, then it's the same for us. Introduction. Also nice to note that Luc van Hijfte has been elected and named member of the Académie nationale de pharmacie in France for his contributions to pharmaceutical developments. And thereby, without further ado, I think you'll all agree that he's the perfect person to guide us through the first part of this webinar, in which he'll share his vision for the drug discovery engine of the future, as well as how public-private partnerships can contribute to developing better drugs faster. So Luc, the floor is yours. And again, if you just, if it looks like full presentation mode to you, then it will look the same for us. We still see your bottom bar now.
Yeah, well, I don't know how to get rid of that one. On the other hand, I think it will not harm. So again, for the sake of time, let's step into my presentation. It's really nice to meet you all, although I cannot see you. But I hope I will indeed give you some ideas on the future of developing small molecule-based cancer therapeutics. Okay. Oh, here we go. Can you see this all correctly? Because seemingly, yeah.
There.
Here we go. I will first of all say a little word on, I've been in this business for already quite some time. And there has been a real transformation of pharmaceutical R&D ongoing that is really towards open innovation. And that's an opportunity for Oncode Accelerator. I will also talk about the importance of collaboration between academia and CROs and give you an example, a case study from Oslo University and Symeres that we developed with tankerase as a therapeutic target for oncology. Then I will explain a little bit about the path from lead to clinical candidate and use that then to come to what kind of the future, how to improve, how to accelerate, how to do things faster in the drug discovery process. Now, when I started my career, which is about in the midst of this slide here, which is indeed already quite some time ago, at that moment, pharma started seeing that the cost, the expenditure for bringing new NMEs to the market was kind of started to increase a lot, where the number of NMEs that were coming out was not increasing. And this, I would say, became, I would say, towards the end of the last century, kind of dramatic. And then there were these big mega mergers that I think had a negative impact. But in the meantime, there has been, I would say, a shift in the way on how the pharma R&D is looking into doing drug discovery. And that has to make with what I tend to call closed innovation. You may know that it is a very long, risky process to get from a starting discovery project to bring one product to the market. You may count somewhere in between 12 to 15 years to make the whole process, but even furthermore so, one needs 50 discovery programs to eventually arrive with one product on the market. And this is something that by itself also needs improvement. I'll come back to that later on in my story. But now, when I started my career, and that was in '86, at that moment, and again, it's schematic, all this work, all the work that needs to be done was done by internal research at large pharma. At that moment, there was not talking of, I would say, any type of outsourcing of working in partnership. That was just not existing. And now what has happened is that this has now kind of evolved to a completely new pharma R&D ecosystem based on what we call open innovation. And what has happened also, and you may have all seen that, is that big pharma, they have been reducing footprint. And to some extent, they focus today more on already later assets, clinical programs. That's where the big money is needed. But there is a complete new ecosystem that has been built around the big pharma that consists out of academia, scientific networks. Then you have the biotechs and also the external research capabilities as Symeres, for example. And so when this gradually, these kind of other kind of entities came in to the value chain of the big pharma, they of course, there were a lot of, I would say, um, companies and academia, scientific networks that are present there. And all of them, at the start, they first of all try to reach out to the big pharma and try to collaborate. But what even has become even more important, to my belief, is the fact that these different entities, instead of looking into the big pharma, they really started collaborating together to bring their respective expertise together. And I would say to kind of come up with likely kind of the drugs of the future. And so this is something that happened. And last but not least, also, another important fact, of course, this is costing a lot of money. And at that moment also, I would say the venture capital money, the funding agencies has become very important to kind of also ensure that the projects get through the pipeline. Now, to kind of highlight a little bit on how could this work and what this could give, it's something that is not related to Oncode Accelerator. It's a project that we've dealt with in the past through a collaborative effort with Oslo University Hospital and ourselves, and where we've been successful in helping Oslo University to get a nice grant from which we at that moment could get paid with some FTEs and really take this project on. So it's kind of project, again, together with Oslo University. Now, the people here are very important people. This is Stefan Kraus and Jo Waller from Oslo University. They're already since 15, 20 years, they are looking into any type of targets that could be of importance for cancer. And something that has been clearly described is that the WANT and somewhere related to hippo signaling pathway, that they are very important for, I would say, the tumor type of tumorigenic activity in the body. So they were very, very interested in trying to find kind of starting points for the range beta-catenin signaling. And for that, and again, was kind of done purely here at an academic level. Symeres was not involved at that moment. So Stefan and you, they did do kind of a medium throughput screening, looking into some kind of a direct kind of a functional type of assay, looking at indeed kind of the catenin kind of downgrading. And they found interesting hits. And to give an example of one of these hits that afterwards, they needed kind of, I would say, further profiling, further identification. They, of course, needed to be looked at in different efficacy measures that could be done. There was also some kind of test that Stefan and you were doing to show specificity. This is a Xenopus model in which it is known that when you administer here WANT8, that you kind of get a very special phenotype, and that could be reverted with the compound that they had observed here. So really showing that this was really kind of working on the WANT beta-catenin signaling. Last but not least, also very important in any type of project that is being run, they started fishing after which target this could be, and they came to what indeed proved to be the real target, which is Tankerase 1 and 2. Tankerase is a member of the PARP family of molecules, which is poly ADP ribose polymerization proteins. Some of the PARPs today, even PARP1, for example, has compound already on the market. Tankerase is different, but what these compounds tend to do is they tend to be involved in kind of regulation of protein levels in the body, because they will, through parcellation, as we call it, and ubiquitinylation, they will define which proteins need to be destroyed. Now, a bit of biology. WANT beta-catenin activated is leading to gene target transcription. It's a very important target, and so WANT beta-catenin is involved in there. So high beta-catenin levels, high gene target transcription. Now, beta-catenin by itself is regulated through what is called a destruction complex, with one of the core proteins in there, axin. And so this destruction complex will parcellate, ubiquitinate beta-catenin, and at that moment get degraded. So high levels of this, low levels of beta-catenin. Tankerase indirectly is involved in the control of axin levels. So active tankerase will at that moment, active tankerase will lead to, I would say, kind of lower axin levels and high beta-catenin, but inhibiting tankerase then will lead indeed to a downregulation of the Wnt beta-catenin cascade. So this is kind of to the biology, and that is where then it kind of fits into kind of any anti-tumoral activity. Now, you may know that the process of taking a hit, and especially on an academic setting, and also afterwards with the means that we had available, it has been a quite lengthy, tedious project to, I would say, convert, this initial hit, which is GW74, to these compounds OM153. And Yuval, Stefan Kraus, together with University of Oulu and with FMP in Berlin, with Marc Nazaré, have been able to take the project on up until OD336. And that's where they realized that they needed, I would say, industrial help, because they realized that they could not get all the parameters right, that they really need assistance there. And that is where then the team from Symeres, with especially to be named Anita Wegert here, with Ruben Leenes, which was the project leader, have done an extensive job in at that moment, starting to build SCR, structure-activity relationship, around this molecule here, understanding what's wrong, what parameters do you need to optimize to get first to a first kind of advanced lead compound OM1700 to eventually land onto these new compounds, these new entities. They gave us something very interesting though, is because thanks to their kind of screening, they had come up with compounds that, unlike the first generation PARP inhibitors that were sitting in a nicotinamide binding site, they were, in this case, we were binding here in the adenosine binding site, which allowed a high selectivity. And that also indicates that GO7LK, for example, one of the initial lead compounds they had, was extremely selective for tankerase. So very interesting, but still, and also another thing that should be mentioned is that also this GO7LK in a colorectal cancer model was showing already kind of very nice anti-tumoral type of activities. But as soon as you wanted to go to higher levels, higher doses of the compounds, you also could start seeing is that there was a severe body weight change, and that is always linking to, I would say, toxicity. And so there was something, I would say, again, highly selective compounds, but still, due to the fact that the overall parameters were not right, we needed to dial out the toxicity in there. On how does one step into this? Well, this is something for a medicinal chemist, which is called a design make test cycle. You have kind of hits, you have chemical matter that you start from. You will start kind of making changes into these molecules at different regions. You try to understand on how these differences then is impacting not only the activity, but also the physical properties, the ADME profile, even the toxicity. And you go gradually through iterative optimization cycles in which you try to optimize your molecules. And to some extent, I always compare this to kind of trying to solve a Rubik's Cube. You should not kind of think about one phase at a time. You really need to take all these parameters on board during the overall, I would say, kind of going down the line. And again, as you will see, this is something where we do believe is that the augmented intelligence, the artificial intelligence, the computer sciences will ultimately kind of also helping us a lot in making these steps. So again, so our medicinal chemists indeed they took OD326 and they started to go through design, make test cycles, starting to optimize parameters to come eventually at a better molecule that you can see to the right there. Has taken some time to eventually land there, but again, coming back to trying to solve the Rubik's Cube, so the compound OM153 profile. So what we normally tend to do is we set up something which is called target product profile. This is where, based on all the different activity, DMPK, safety, all these kind of parameters, we want to get them right. And what we know, the ideal compound never exists. We advance step by step and we try, and we had here brilliant activities, very nice activities. DMPK, there were some subtle things that were not right, but I would not call them red ones. This by itself could already be a clinical candidate, but to some extent, as you will see, we have been able to also further improve on some of these DMPK parameters. Now, of course, with this kind of better molecule at hand, what was kind of helping us then was, first of all, we looked again at, do we kind of see again a very nice anti-tumor type of activity, and we could even see is that even we saw kind of even activities even up to the 0.33 milligram per kilogram level, which is very low doses to get, I would say, remarkable results. But even more so, and that was really very interesting in this project here, is that even up to the highest levels, the highest doses that we were giving then with these type of molecules, that we did not observe any, I would say, impact on the body weight. So already showing that we here had shown that there was a therapeutic window, that we had been able to dial out all the toxicity. In the meantime, also OM153 could be nicely detected and dosed in plasma and in tumor, again, also leading to really kind of make the relationship between, I would say, our biological target and the anti-tumor activity that we could be observing. So again, so this was for us, of course, an incredible nice story. This is something where we've been able to take a lead molecule to bring it to a clinical candidate status. So what's now awaiting us, I would say, is to do the same type of activities now for Oncode Accelerator. Now, again, having said that, the profile was not yet perfect. So we want to solve all the phases of our Rubik's Cube. And that is something we've been able to do in next generation type of molecules, in which we got to a molecule that really has a very, very nice overall profile. And especially, I would say, on the DMPK front, where you can see is that still our kind of first kind of compound, the OM153, was not yet showing a very nice, I would say, PK concentration time curve. But you could see here that the molecule, both I would say IV and even per oral, was giving us extremely nice, I would say, PK traces. So that means that kind of we really could tell here that we had kind of accomplished our kind of what was expected from us. Now, having said all this, kind of on how can we get better at we already do today? To some extent, kind of once someone has a new target, you will try to find hit identification means. I will not go into the details, but you need to start at that moment to get to somewhere chemical matter to start from. You will get into design make test cycles. So of course, the choice of the target is extremely important. The choice of the target is extremely important because if the target is really well chosen from the start, we can only hope is that we will not need 50 discovery projects to end up with one project at the end, is that we can maybe start even with 20 discovery programs if the targets are well chosen. What is also important is to reduce the attrition, but also to reduce the timelines. And to reduce the timelines, at that moment, we also need, I would say, better predictive models. We need indeed better targets, better predictive models, and to some extent also, we do need faster SAR building. And that's really what Oncode Accelerator is about. It's really about bringing the patient cohorts and the information on the patients closer to where we start from. It's also to have the organoids as even better predictive models to kind of be able to really kind of have better activity type of kind of readouts, and also the faster SAR building, in which we do believe that computer sciences and artificial intelligence will be helpful. So in the meantime, this is all done by building bridges. And I think with that, I have to thank you, and I want to give the floor to Anthe to tell about the impact of artificial intelligence.
Thank you very much for this very informative talk, Luc. And indeed, it's now time for us to proceed to the next presentation here. So Anthe, if you'd like to start pulling up your slides, I'll give a brief introduction. So Anthe Janssen is a principal investigator at Leiden University and is indeed a medicinal chemistry expert from a different point of view. And his expertise bridges medicinal and computational chemistry. In 2020, he founded his own research group at the Leiden Institute of Chemistry at Leiden University, which focuses on developing and bringing useful AI tools to the chemical biology workbench. I think Luc's talk set you up perfectly, so rather than take any more time, Anthe, the floor is yours to tell us more.
Thank you so much, Alex. And indeed, Luc, thank you very much for preparing the crowd and introducing the topic as clear as I could possibly wish it to be introduced. So yeah, my name is Anthe. I'm part of the Oncode AI platform of innovation. I'm involved in two of the work packages there, and I hope to show a little bit on how we think AI might help the future of developing small molecule-based therapeutics along. And now, of course, the slide has to continue. So for today, I think this is exactly what Luc also sort of implicitly defined. When I talk about a drug, I talk about small molecules that bind a protein and thereby blocking or altering its functionality. So be it a receptor ligand, an enzyme inhibitor, but it's the sort of traditional small molecule effectors that we sort of know and love. And then again, this pipeline was also showed in a different shape. We are in the AI field currently, at least the work that I'm doing, mostly focused on the preclinical work, so the lab work. And there we are focusing on the hit finding and lead optimization stages. So basically the design, make, test cycle that was just on the slide, how we enter that with a hit, and then how we proceed through that cycle as fast as possible. And I think that is where a lot of the small molecule AI is currently focusing. So how do we think about doing that using AI? And I think the first thing that we should ask ourselves if we want to answer that question is to think about what AI actually is. And apart from a word or a buzzword that you hear basically everywhere nowadays, it kind of gets boring even if you work in it. The term artificial intelligence is, I think, used very generously for a lot of things. And let's just get on the same page on that. So artificial intelligence is actually a very broad definition. Even a small chess player board that you carry around in your pocket that plays very well chess, and at least beats me every time that I try to beat it, is technically considered artificial intelligence. It's very narrow intelligence. Don't ask it about the weather tomorrow, but it is mimicking human intelligence in a way that basically it's better at this particular form of intelligence. What the news is actually all about is about specifically the deep learning, machine learning based neural networks, artificial intelligence. And that has nowadays more or less become synonymous with ChatGPT and all the other image generators that you see out there. And all of these, in the essence, are built on neural networks that try to sort of mimic human intelligence also from a programming point of view. And it's these technologies that we try to use in our field of research. And maybe the question is why? Well, probably because we aren't smart enough. But the real reason, I think, is that we are looking for drugs. I think drug discovery is one of the best sort of definitions that you could have. We're really out there looking for the best molecules to use. And AI can help us with that. If you look at the current number of drugs, which is somewhere in the order of magnitude of 10 to the third, that's a surprisingly small number of molecules that we actively use to cure humans. Especially if you compare it to basically more or less standard screening deck that the pharmaceutical industry uses, which has at least 1 million molecules. And definitely if you compare it to all molecules that we think are drug-like, which is at least 30 orders of magnitudes larger than what we currently use in the clinic. So there should be way more molecules that do amazing things. We just need to know how to find them. And on the target side, we currently treat diseases by targeting about, let's say, several hundred protein targets. But there are over 100,000 druggable proteins out there. If you include all the bacteria and all the isoforms in humans, there is a significant wealth of targets still out there to be discovered. And if you then map all of this against each other, there's an immense and truly unfathomably large unexplored chemical space that we as drug discoverers are sort of slashing our way through this jungle. And I think any help that we can get is welcome, and therefore machine learning, AI, however you call it, is happy to join the search party and try and find our next drugs. So what might that look like? Well, I think a lot of you in the audience will recognize this layout. What if we could just type in a question in a website where we just say, today I want you to design a drug targeting this protein that I'm interested in. For today, this will be fatty acid amyloid hydrolase. This system, of course, doesn't exist yet. I don't know if we'll ever get there, but ChemGPT that will just generate you drugs whenever you feel like it won't be around for the next few years, I think. However, we are working on basically the sort of first steps of this system. And the way that we try to do that is by using a technology actually developed here in Leiden as well in the group of Gerard van Beste, who is also part of the AI platform, which is called DrugX. And this is a molecular generator that allows us to generate molecules never seen before. And I say generate, I should say sort of in silico generate. This is not a synthesizer. These molecules don't physically exist. And we aim to basically immediately generate the perfect molecule. So we try to skip Luc entirely, don't go into this design make test cycle, but directly go from some kind of hit, some idea into the perfect molecule that should go into the clinic. Don't worry, Luc, we're not there yet. So how do we do this? We start basically from a language model. And this language model is trained on millions of molecules from literature to sort of learn the language of what a molecule might look like. Now, after this training stage, this model will then sort of speak the language of molecules. So if you ask it, give me 100 molecules, it will happily do so, but these will be more or less completely random. And then the hard part comes, how do you get it to generate the molecules that you want it to do? And then the image immediately becomes way more complicated. So we pre-train this on just molecules. And currently what we do is we then enter a stage which we call fine-tuning, where we show the model hundreds of examples of molecules that we know interact with this protein in a way that we want. So we basically bias it towards the molecules that we think, based on literature, based on in-house data, should do the trick. After this fine-tuning stage, the model then generates molecules that look like the molecules that you think will interact with the protein, and in this case, inhibit it. And then we enter the final stage, which is called reinforcement learning stage, where we basically give the model the task to generate the best molecules it can based on some kind of scoring criterion. And the model will sort of play a game against itself where it's continuously getting a reward or it's getting a slap on the hands if it's doing wrong. And it will improve and it will learn on which molecules it gets a good score on. At some point, of course, this saturates and you have a final model that if you say generate 100 molecules or 100,000 molecules, if you feel like it, it will happily do so. And these molecules will all do very well in the scoring criteria that you give it. And in this way, we can now generate virtual libraries of thousands of molecules that have attractive chemical properties and should interact with our target. Now, how do we score this? Because this, in the end, is one of the key points in this whole loop. Traditionally, we use a QSAR model. So this is also a machine learning model that tries to predict the bioactivity of this molecule it just generated. But we couple this to what we call the quantitative drug likeness parameter, which is a weighted metric of molecular weight, lipophilicity, and others that sort of try to predict all these parameters that Luc showed that should meet a certain criteria to be drug-like, to be soluble, to be administrable in a favorable way. And these two parameters together, just two sides to this Rubik's Cube, so it's a way simpler problem, are what this model tries to optimize and generate new molecules for. So an example from our lab, we performed a medium throughput screen, found some hits. One of the hits is on the slide. And this hit has a micromolar potency for the target at hand. And we went through this whole training loop and ended up with, in the end, 1,000 generated analogs. We docked all these, we ranked them on affinities. And in the end, we selected the two analogs shown on the slide for synthesis. We now have them in hand. We're not as fast as Symeres in this. And we still need to do the biochemical evaluation. I was hoping to get that through before this webinar, but unfortunately, lab cleaning activities at the end of the year prevented that from happening. So I can still say positively that these will be highly active without being proven wrong yet. So this synthesis and evaluation is definitely part of the assignment in order to show that this system actually works. We're not just predicting molecules that are active, we're trying to make them as well. So overall, before we actually have the bioactivity data, is this approach any good? And I think it can be. But there are some downsides. And the major one that I see in this approach is that because you basically bias the model based on knowledge you already have, you will generate a lot of lookalikes. You need quite a bit of data before this model can start to generate molecules that you think are interesting. And you also can only rank them based on the QSAR, which again needs data to work. So you first need to make hundreds of molecules before you can start to generate these new molecules that you didn't know. So there will be a lot of Me Too drugs that you can generate in this way, but truly novel chemical matter is difficult. So my question then is, can we do better than using all this data from literature? And can we really go de novo? And this is another project that we have been working on. And for that, we try to use a tool called docking. Now, docking is definitely not AI. This is a technology that's been around since the 80s. There are now, of course, AI models that do docking, where basically what we do is we try to find the 3D fit of a small molecule inside the protein. And by fitting inside this protein, you indeed get the effect that you want. And this is a technology that's been around, so this is quite well optimized. It's fast, it's easy, and it works quite well. And overall, we can say that the finding of the 3D structure is quite good. However, how tightly this fit then binds, so what is the binding affinity, the chemical energy that is associated with this interaction, that is apparently, based on 40 years of trying to figure this out as a science, that's a real challenge. And the problem, I think, is a lot to do with data. The protein data bank has thousands and thousands of complexes, but for most of these, we don't know the biochemical activity. And vice versa. There's a lot of biochemical data in, for example, Campbell and other databases, but for those, we don't know the structure. So if we want to use artificial intelligence, we kind of have to squeeze these two balls together to get a decent sized data set that we can start training our models on. So what we realized is that if docking is so good at predicting these poses and all this biochemical data is out there, we can generate this predicted data set where we use artificial data to train artificial intelligence. So that's what we did. We started by gathering literature data. There's a very nicely curated biochemical data set out there, also from Gerard van Wester's group called Papyrus. There's from another group at Oncode Accelerator, from Perakis at NKI. There's Cliffs, which has a lot of crystal structures, nicely curated. And these all relate to the same protein family, the kinases. So with this data set in hand, we now have more than 200,000 protein structures that we can start to play with. And if we then look at the state-of-the-art prediction models, we can see that indeed predicting this affinity value is difficult. You don't need to be an expert in statistics to see that a correlation coefficient of 0.0 something is probably not going to tell you much. So we need something better than this. And the way that we did this with all this new data is to abstract features from these 3D poses and basically feed these into a very simple neural network. This is basically as simple as they get. So a very small 3-layered neural network that uses features from the 3D crystal structures to just tell you what the affinity of that molecule for that protein is. And we have the literature data, so we can train this model to predict the right values. Now, does this approach work? Well, yes, obviously, otherwise I probably wouldn't be telling you this. So what we can now do is, with a relatively high confidence, for about 40% of the kinases, we can predict the biochemical affinity just by docking the small molecule into the protein structure. So you don't need to make the molecule, you don't need to measure the bioactivity data, you can immediately tell what the affinity value will be. Of course, the performance varies a little bit per kinase, but overall we see quite good performance. So good that we decided to build a small software tool around it, which we called Kinase Docker. This completely automates the whole docking procedure, the whole machine learning procedure, and you get these 3D views of the poses in order for you as a medicinal chemist to assess whether you actually like the pose that your prediction was based on. And this is released currently online, completely free to use. Offline, so you can use this on your own computer, not sharing your structures with anyone. So what is then next? I think we still need to be faster. This docking works, but it is relatively slow. And of course, we always need better models. 40% of kinases is not 100% of kinases, and is definitely not 100% of all proteins. So we need to be better at this. And when I would really want to combine this with these compound generators so that you can really just shop around. You choose this very well validated target that Luc talked about. You look up the AlphaFold structure, you run this compound generator, and you get structures of small molecules that bind it with high potency, high oral bioavailability, low blood-brain barrier penetration, if that's what you prefer, etc. And who knows, when ChatGPT came out, nobody really expected that. So maybe ChemGPT is actually a lot closer than we think. I would love to be proven wrong in this. So of course, I did not do all of the work. I have to thank a few people that are on the slide from our own group, Molecular Physiology at the LIC, at the Computational Drug Discovery Group at the LSCDR, also here in Leiden, and the Informatics Department, Oncode Institute, and of course, Oncode Accelerator for making all this research possible. And with that, I happily give the floor back to Alex.
Thank you very much, Anthe, as well as Luc, for these really fantastic presentations. I think you gave a very good overview, not only of your own fields, but also how they work together. Thank you for that. It's definitely in line with the goal that we have with these webinars to build bridges across topics of different work streams and platforms. I invite everyone who's watching to start typing in earnest in the Q&A section if you have any questions. I don't see any popping up yet, so...
Alex, may I chime in?
Absolutely, that's what I was hoping would happen.
Because during the talk of Anthe, at some stage he wanted to kick me out, or I don't know what the meaning was. And that reminded me on a very, very nice discussion I had with Mark Mirko. Some of you may know Mark Mirko from Boston area, quite well-known biotech entrepreneur. We were discussing about AI, and Mark, you said is that they have done an experience. You spoke also about the playing chess, and we know that today the grandmasters, they know today they cannot win anymore from the computer. But now the experience they've done is they combine the grandmaster with a computer that was trained to play chess against a computer by itself. And the computer by itself cannot win from the two, meaning that this may evolve, but Luc is not kicked out yet. It's going to be, I think, a very good, I would say, connection between the human intelligence and the artificial intelligence that will, I think, make the biggest impact. Just a little note that I'm not out yet.
I can only wholeheartedly support that that as an idea. I think the combination of human creativity and the immense knowledgeability that these models can achieve, that is where the power lies. And making use of our time, which is expensive and I would say valuable, as best as possible by sort of being able to bounce ideas off, in this case, a model, getting, for example, a solubility parameter, or so would this work? Well, probably not for whatever reason. And this is also why, for example, this graphical user interface is one of the things that I strongly feel we should do as a sort of AI developers. If you want to be well, if you want to have impact, which is nowadays a sort of a curse word in academia, you should have people use your tools. So you need something better than just some kind of GitHub repository. You need something that people can play around with, that they can test and that they can use. And if that's not there, then yeah, you made it for just the paper, and that's not what I'm here for, at least.
Good to hear that humans still have a chance to contribute to biology. On this note, we have a question from one of the participants. Does AI actually understand the chemical and biological interactions of the molecule? Could you tell us a bit more about how this actually works in practice, Anthe?
That's a very deep question, I think. At this moment, I'm inclined to say at least most models definitely don't. Most of them are very sort of task specific, and in the end, they are just statistical sort of recognition thingies that discern trends that might be very hard to discern for us. So in a way, a lot of what these models are is just fancy QSAR models, which is cutting them really short, but still in essence, I think this is true. But same as, for example, for the advanced ChatGPT and the like, you can see some emergent properties sometimes. And I think there is more and more large AI models that combine multi-objectives, multi-tasks, and that then seem to be able to sort of use information they learn from one task and start to use that in other segments of the questions as well. And yeah, that's where I think maybe understanding becomes a thing. But I'm not a philosopher, so I would have to pass this on to someone that's more knowledgeable in that area.
Got it. Next time we need a philosopher as a third speaker for the end of the webinar. Thanks for the answer, though. In the meantime, I would like to remind attendees before they leave to please fill out our survey. Saskia has placed it in the chat, so we'd really love to hear in a 5-minute survey your feedback. This is not to say we're ending. We have plenty more questions. The next one is for you, Luc. One of the participants asks, how many compounds were initially made in the project? Or actually, I believe he means over the period of the project, how many different compounds were made within the use case that you shared? And did you determine all parameters for all of those compounds?
So first of all, on how many total numbers? It's an order of magnitude. I know at Symeres, and again, this was not a very extended team. This was like 1.5 FTE over like 18 months. I think we made something like a few hundred compounds. And before that, when it was still in the hands of the FMP in Berlin with Marc Nazaré, honestly, there I do not know. But again, I think today you can count on the fact that to get to a clinical candidate, that people will make something in the 500 to 1,000 molecules, which is in the same range. And the second question was, do we determine all of the parameters on all of the compounds? The answer there is no. There I can relate it to playing mastermind. You do it on a sufficient number that allows you really to show that there are trends. And some of these parameters you don't need to. And again, not that I'm going to say something, because there's, I think sometimes, and that's at big pharma, they have processes. You make a molecule, these go into a process, and they kind of get checked across all of the different parameters, all of them, which for me is a bit of a waste of time. I think you need to do it in a smart way. You shouldn't do it on all of them, but whenever you need it and you want to optimize some parameter, then you need to zoom into that one, while kind of ensuring that all the rest, all your other phases of your Rubik's Cube get along well. So, but no, not on all of the molecules. I think you need to do it as smart as possible.
Thanks. So indeed, an element of prioritization throughout, clear. As a follow-up question to that one, the same participant asks Anthe, would that be enough compounds, or how many compounds would be needed to in order to generate or make to the point at which AI would be used or useful?
Yeah, that's again good question of Paul. I think with a few hundred compounds, the technology that I showed now, and there's not just one form of AI, but the DrugX tool that I showed in the first half would need 100 or a few hundred compounds to reliably be able to do this this kind of extrapolation. You would need to make these, and you would need to test these. And to sort of counter Luc there, us a data-hungry AI specialist would love all compounds to be tested in all assays, because we need this data in order to be better. So there's a lot of biochemical data out there that can help us get you through the first stages. But when it really comes to lead optimization and trying to improve solubility or membrane permeability or whatever, these things are so rarely measured and also rarely published that we struggle as a community because the data just isn't there, or at least not publicly. But so I completely understand that you don't have the time and money to measure everything. That's the other side.
Yeah, this ties into the next question from a participant, actually. So clear that you need positive and negative data. And within that, is there a specific type of data that would really be required in order for a drug discovery project to benefit from the AI approach that you have shared? So must there be functional data, binding data, both structural information? Can you comment?
Yeah, so the first part that I showed didn't use actual structural information, so just the chemical information was used. You see more and more that, especially with AlphaFold out there, that structural information is sort of taken for granted and taken along. And this primarily focuses on binding data. So this is just the molecule binding to the protein. You could replace this parameter that gets optimized with whatever. So if you have cellular functional readouts that you can in high numbers generate, that of course would get you better predictive models that Luc again referred to. So then you sort of skip the first step where you just try to make something that works really well in the simplest of systems, but gets you to actual potent molecules in cells. And that's just what do you learn on and which data do you provide the model to try to mimic and optimize.
So the data really has to match the question that you're trying to answer with the AI, if I understand correctly.
At the moment, there's a lot of people that say AI is just an abbreviation of advanced interpolation. So if you don't give it the data, then it really won't be able to do much.
The next question from another participant is for you, Luc. So he asks, he's clearly knowledgeable about the case study in which you showed. He asks, from his recollection, XAV939 and IWR1 toxicities were linked to TNK inhibition. So how did you rationalize the difference in toxicity profile with the compounds that you optimized leading up to OM0815? And was that a matter of selectivity within the TNK family or off target from unoptimized TNK inhibitors?
Okay, well, first of all, maybe something that nobody here will realize is that I have to say bonjour, Cédric. It's really a pleasure to get a question from a collaborator of mine of about 25 years ago, I think, close to. So Cedric, to some extent, yes, indeed. GI toxicity was one of our big concerns in this. What I should mention is also that G07LK has also been published, the work by Genentech also showing indeed GI toxicity. So one of our kind of more important things we looked upon with our new molecules was indeed GI toxicity. And to some extent, we've gone into more extended tox studying, and we have not been able to at that moment really show any of that. So to some extent, yes. So then your question is coming then, and that is then where to some extent we have to I'm not so sure to have the real answer. We have an extremely selective compound. We have really on the tank array, so meaning that likely some of the other molecules, they may have had some other problems. To some extent, even one of the things is that also I think we have been working on solubility. Some of these kind of GI toxicities to some extent at higher doses, but then if you have highly insoluble compounds and if they would kind of tend to drop out in your intestine at that moment gives you kind of local higher concentration and that's leading to GI toxicity. Honestly, I do not have a kind of a full answer to your question, but indeed it was one of the bigger concerns we had in this one, and we've been able at least to show that therapeutic window could be obtained at least in the cases that we've shown here. Yeah.
Thanks for the detailed question and detailed answer. And perhaps interesting looking back on that case study, would you have seen a point at which AI would have been very helpful in developing that molecule together with Oslo University?
The way I'm looking at this today, it's human intelligence to define when you go to your design make test cycles. You will design a few molecules where you make some changes, where you try to address some items, can be some kind of permeability, can be solubility, just name it. You bring them through. And each time the chemist is thinking about a number of options that he will work upon. And if you would have already have a tool by the computer that can help the chemist to define or to kind of take maybe the best options within the different options that he created in his mind to go for these, and if then the computer is true, at that moment we would be just getting more efficient in getting through design make test cycles. So this is something where, for example, I already see kind of the importance of AI. Another thing where I see the computer for me, and that's for me straightforward, if I give a list of 1,000 molecules and 3 parameters to a missile chemist, he will spend a day to try to grasp some kind of trends in there. If you can learn a computer to look in the right way at these data, a computer will not be stopped at 1,003. He will be able to cope with 1,025 parameters. So I would say that's the strength of the computer kind of, but we really need to learn the computer on how to look in the right way onto kind of the data it's receiving. And that's a little bit kind of also the design mix test cycle, the incremental growth based on the data to get these new models better and better in their predictive mode. And that's what I think, have to have a look at.
Thanks very much. With that, we need to wrap up. I see one question I would love a yes/no answer on Anthe. Do public-private organizations like Oncode Accelerator enable research groups like yourself to go from unpublished data from industry as well to improve your model? Yes or no?
To some extent.
To some extent. Okay, that's a good note on which to end. So from here, I would like once again to thank all participants for attending. Please do share your feedback by scanning this QR code or navigating to the link that Saskia shared earlier in the chat. And of course, a big thanks is due to both Luc as well as Anthe for sharing your expertise and kindly giving your time to do so today. I see some applause from the participants. I think it was much appreciated. With that, I would like to say one more time, please do follow us. Both Symeres as well as Leiden University are partners within Oncode Accelerator. So we'll be sharing more about them as well as our other partners over the coming year. And this is a good point at which to wish you all happy holidays, and we'll see you back in 2025. Thanks all.
Thanks all.
Thank you.
Bye-bye.